Lesson number 10, Infections of Body Surfaces. From today's lecture, we start talking about infections that affect different parts of humans' body. And as I said, today we're going to talk about infections of skin and mucous membranes. When you study this lecture and the rest of the lectures of this semester for your future exams, please pay attention on a few things. Number one, when you study each infection, you have to know to what group of pathogens the pathogen that causes that infection belongs to. If it's bacterium, if it's virus, fungi, or protozoa, etc., etc. Second, you have to know causative agent for infection because certain symptoms of infection are caused by pathogen, but some symptoms are caused by toxins that are produced by pathogens. So you have to pay attention what is the causative agent for each infectious disease. And the last one, if infection is caused by bacteria, you have to know Gram-Stein reaction, morphology, and arrangement for that bacterium. Alright, and today's lecture will start with anatomy of the skin. Our skin is composed of two main layers. Those are epidermis and dermis. Epidermis has two main layers in the structure. Those are stratum germinativum and stratum corneum. Stratum germinativum is a deeper layer of epidermis, and this is when new epithelial cells are made. Stratum corneum is the surface layer of the skin. It's made of several layers of epithelial cells, and also it's covered with a waterproof protein. We call it keratin. You have to remember that epidermis is the area of our skin where infection usually happens not very often. And the reason why, because the epithelial cells of epidermis are very close to each other, very tight to each other. The deeper layer of our skin is called dermis. Dermis. It contains blood vessels, lymphatic vessels, nerve endings, and cells in dermis are not as close to each other. Plus, because it has a lot of lymphatic vessels and blood vessels, every time an infection gets into dermis, it spreads quickly, rapidly. So how usually infection gets into the dermal area of the skin? The main openings for infections will be opening of the hair follicle and the sebaceous glands. So how is our skin protected? Sebum and perspiration contain a lot of enzymes, we call them lysozymes, and those enzymes are toxic to a lot of pathogens. You probably remember from previous lecture that skin is an area or site of normal biota, and the greatest number of microorganisms are usually found in the moist areas, under arms, in the groin area, around the nose and mouth. On this slide, in this chart, you see a lot of terms that we use to describe lesions on the surface of the skin. You have to remember that skin infections actually described by their appearance. And for your future other classes, including microbiology class, I would recommend you to memorize those terms that we use to describe lesions on the skin and mucous membranes. For example, erythema. Erythema means redness of the skin. Petechiae means small, tiny purple spots on the skin. Purpura, it's a large purple spot. So please go through this chart and memorize all terms you see in this chart because we're going to use them a lot. And we start with first pathogen that can cause infections of the skin, Streptococcus pyogenes. Streptococcus pyogenes is gram-positive coccus in chains. It is catalase negative. It can cause different infections of the skin, and actually the type of infection depends on the depth of infection and virulence of the strain. We start with the surface infection that affects surface of the skin. It is impetigo. It's called impetigo, and this is the most common infection that usually can be seen in children. This infection starts as a tiny break on the surface of the skin, and then that lesion fails to heal, grows, becomes larger, and eventually the clear liquid starts oozing through from this lesion. Eventually that fluid or that liquid dries out into what we call honey-colored crust, and that crust can get broken again and it starts oozing again. If you culture that liquid that comes out of that lesion, besides Streptococcus pyogenes, you almost always will be able to find Staphylococcus aureus. Next streptococcal infection of the skin is erysipelas. Erysipelas affects deeper layers of the skin. It can affect epidermis and dermis, and therefore it spreads quickly. Can get into the lymphatic circulation, into the bloodstream, and become eventually systemic infection, which is a life-threatening condition. So the treatment of erysipelas must be started as soon as possible with antibiotics. The next very serious infection or condition that can be caused by Streptococcus pyogenes is streptococcal gangrene. Another name of this infection, you probably heard about this infection as flesh-eating bacterium or flesh-eating strep. So basically, what happens to the tissues when they are affected with streptococcal gangrene, they start melting. So why is this infection so aggressive? Because Streptococcus pyogenes has ability to make a lot of very aggressive toxins, and those toxins cause very severe or intense inflammation. Inflammation can be so intense, so severe that blood vessels are destroyed. And of course, when blood vessels are destroyed, overlying tissues, overlying skin dies. Streptococcal gangrene affects epidermis and dermis, and because it can quickly reach dermal areas and then from the dermal area get into the bloodstream, this infection is extremely life-threatening, dangerous, and antibacterial treatment must be started as soon as possible. So the question is, why Streptococcus pyogenes can be so aggressive? What are the virulent factors of Streptococcus pyogenes? Number one, Strep pyogenes has ability to produce what we call M protein, surface M protein. And remember, we already talked about surface M protein. I told you it interferes with phagocytosis. As I said, Strep pyogenes also produces different toxins, very aggressive toxins, and what they can do, they can kill white blood cells, they can dissolve blood clots, they can destroy red blood cells, and the main thing, they actually dissolve the substrates that keep epithelial cells in the skin together. That's why tissue basically starts melting. Also, those toxins can provoke immunological mediated tissue destruction and hypersensitivity reactions. For the treatment, we're going to use antibiotics as soon as possible. On this slide, you see two pictures. This picture on the left shows you a honey-colored crust of impetigo, and on the right side, this is a patient with erysipelas. The next pathogen is Staphylococcus aureus. It is gram-positive coccus in clusters. And today, Staphylococcus aureus is #1 problem in our hospitals. Why? The first reason why, because it becomes more and more resistant. The second reason why, because Staphylococcus aureus has ability to infect any tissue, any organ of human's body, including skin and mucous membranes. Staph aureus can cause different skin infections, and we'll start with the surface layers and then go deeper. So the first surface infection is impetigo. Yes, impetigo also can be caused just by Staphylococcus aureus. It's a superficial infection, but the lesions of impetigo look a little bit differently if they're caused by just Staphylococcus aureus. That lesion looks like reddish, thin-walled vesicles filled with clear exudate. No pus will be produced. The next infection is folliculitis. Those are pus-containing lesions that affect hair follicles. And by the way, the eyelash follicle infection, staphylococcal infection, is called stye. And then we go deeper in the tissues and we have furuncles and the carbuncles. Those are large pus-containing lesions that affect epidermis and dermis. And even larger lesions we call abscess. It's accumulation of pus, and again it affects usually epidermis and dermis. Extremely dangerous infection. The next one is cellulitis. Cellulitis, it is a diffuse staphylococcal infection that actually affects epidermis and dermis, but it doesn't form pus. Does it mean that it's not as dangerous as abscess? No, it is not. It means it is actually as dangerous as abscess. Why? Because, as I said, it affects epidermis and dermis and can get into the bloodstream very quickly. And the next, the last infection that can be caused by Staphylococcus aureus is called scalded skin syndrome. The causative agent of scalded skin syndrome will be a toxin. It's actually called exfoliative toxin. This infection usually affects newborns, young children, and the main symptom of this infection will be exfoliation of the epidermis. Exfoliative toxins make the skin completely peeled off from the surface. As a result, patients develop lesions that look like a sunburn. Lesions are extremely painful, and those children actually cry constantly. Treatment of staphylococcal infections must be started with draining of the pus. If pus is formed as a result of infection, then those lesions must be surgically opened, and that will speed up recovery of those patients. Of course, we have to use antibacterials, antibiotics to treat those infections. Today, 99.9% of Staph aureus strains are sensitive to penicillin. For a long time, the drug of choice was methicillin, and today, we already have new strand developed, which is called methicillin-resistant Staph aureus, MRSA. So against that strand, we for a long time started using cephalosporins. Next bacterium is Pseudomonas aeruginosa, which is a gram-negative bacillus. It is an opportunist because it has minimal nutritional requirements. It can be found pretty much everywhere, in the soil, in the water, humans, GI tract, and basically it is opportunist, and it means it infects people who are already sick or injured. So, as I said, it is gram-negative bacillus. What else we have to know about it? It has pili for attachment. Why is it so resistant? Because in its structure it has capsule on the surface. What else we have to know about it? Pseudomonas aeruginosa has ability to produce blue-green pigment. It means if pus is produced as a result of infection caused by Pseudomonas aeruginosa, that pus is going to have blue-green color. As I said, because Pseudomonas aeruginosa is opportunist, it usually infects or affects already injured patients. And the number one problem today, Pseudomonas aeruginosa causes in burnt patients. As you see from this slide, 80% burnt fatalities are due to infection caused by Pseudomonas aeruginosa. Also, it can cause problems in patients with diabetes, patients with cystic fibrosis. But please remember, Pseudomonas aeruginosa also can cause infections in healthy individuals. And this slide gives you two examples. The first one is what we call hot tub folliculitis. It's a skin infection. And usually patients develop this infection after they use a public hot tub. The second infection is called swimmer's ear. If you swim somewhere in a pond, in a lake, in a river, and next day you wake up with the pain in your ear, this is what we call swimmer's ear, and usually it's caused by Pseudomonas aeruginosa. Because Pseudomonas aeruginosa is a pretty resistant, stable bacterium, those infections are very difficult to treat. And for treatment, today we use combination of aminoglycosides and beta-lactam agents. And the next bacterium is Clostridium perfringens. Clostridium perfringens is gram-positive bacillus. It is an anaerobe. The source of Clostridium perfringens is soil, because all Clostridium species have ability to form endospores, and in this form they can be found in the soil. And also all Clostridium species, including Clostridium perfringens, produce toxins. So please remember for the future, if infection is caused by Clostridium species, causative agent is always toxin. So let's get back to Clostridium perfringens. Clostridium perfringens can cause gas gangrene. How people usually get infected with Clostridium perfringens? People get infected through contaminated wounds, contaminated with soil. Because remember, Clostridium perfringens can form endospores, and this form can be found in the soil. Now, as soon as Clostridium perfringens get into the wound, what is going to happen next? Remember, it is an anaerobe. That means it will try to go deeper into the tissues and avoid oxygen. How does it do that? It starts producing very powerful toxins. Those toxins kill tissues, and this way Clostridium perfringens goes deeper into our tissues. This way how it spreads. Now, why this infection is called gas gangrene? Because as an end product of metabolism of Clostridium perfringens will be gas. This bacterium produces two actual types of gas, hydrogen and carbon dioxide. So basically, if you look at the skin that is actually covers damaged areas, you will see that the skin is very tight on that surface. If you touch it, you will feel that crackling sensation, which means there is a gas under that skin. If there's a lesion on the surface of that skin, then it starts bubbling with fluid, and it's bubbling on the surface once again because gas is produced. Treatment of gas gangrene. You have to remember that gas gangrene is extremely dangerous infection. It spreads quickly, a lot of toxins will be produced, a lot of tissues will be destroyed, killed, and that is producing even more toxins. So We start treatment with high doses of antibiotics. Of course, to neutralize toxins that produced by Clostridium perfringens, we're going to use Clostridial antitoxins. Very often we place those patients with oxygen-enriched chambers, and this way we also try to stop spreading of the infection. But unfortunately, with the best antibacterial treatment, the amputation is the most effective treatment for the gas gangrene. We have to remove all the tissues and stop spreading of the infection. Alright, on this picture you see a picture of patient with gas gangrene. Next we're going to talk about acne. Guys, what you have to remember is acne is not an infection. This is what we call inflammatory disorder. It's a result of a hormonal disbalance. As a result of that disbalance, patients have increased output of sebum. Sebum usually gets trapped in the pores, within the pores. And remember, as a part of normal biota on the skin, we have Propionibacterium acne. I already told you that Propionibacterium acne belongs to a group of diphtheroids, and it is gram-positive bacillus that form a very specific arrangement. We call it Chinese writing. So Propionibacterium acne uses sebum as a source of nutrients. When it uses sebum as a source of nutrients, it produces end products of metabolism, fatty acids. So those fatty acids actually can irritate skin and cause inflammation. As a result, we see it as acne. So how do we treat acne? Of course, everything has to be started with diet. Those patients should avoid sweets, carbohydrates in any form, because sugar actually can stimulate inflammation in a human's body. In their diet, they're supposed to have a lot of green leaves or any other products that contain a lot of vitamin A. For example, eggs, spinach, carrot. Second approach, we prescribe those patients creams or scrubs that remove surface layers of the epidermis, and this way we open pores of those patients. In severe cases, we prescribe antibacterial treatment, and a very often drug of choice is tetracycline. Why tetracycline? Because tetracycline can be secreted into sebum, and also it has ability to penetrate deeper into the skin. Also, remember I told you when we prescribe tetracycline to the patients, we have to let them know that they will become very sensitive to the sunlight. And the last option of treatment will be Accutane. We use this medication for treatment of severe cases of acne. We call it cystic acne. They usually affect epidermis and dermis, go deeper, and as a result of that inflammatory process, a lot of scars will be formed. So basically, Accutane, it's a high doses of vitamin A. What you have to remember is it's also teratogen. It means if you prescribe Accutane to the female patient, you have to put that patient on the birth control pills. And the last bacterial infection that affects skin and mucous membranes will be leprosy or Hansen's disease. It is an ancient infection. We know about leprosy for 100 years. Leprosy is caused by Mycobacterium leprae, which is a bacillus with the waxy cell wall. It belongs to the same group of Mycobacterium species as Mycobacterium tuberculosis, and that explains why those two bacteria have very similar characteristics. Same as Mycobacterium tuberculosis, Mycobacterium leprae grows slowly and has ability to develop resistance rapidly. There is no Gram-Stain reaction for these bacteria, and actually we do not use Gram-staining technique to stain those bacteria because of the different structure of the cell envelope. But we use a different technique. We call it acid-fast technique. What is the source of infection of leprosy in nature? For a long time, scientists had difficulty to grow Mycobacterium lepra in the lab because it has a lot of requirements. Until in 1971, they actually found out that actually the source of leprosy are very ancient, very old animals called armadillos. So today, scientists actually use those ancient animals in the labs to grow Mycobacterium lepra. Why Mycobacterium lepra grows in armadillos? Because armadillos have lower body temperatures. And yes, Mycobacterium lepra grows at the lower body temperature. And that explains why it usually affects peripheral parts of human body, peripheral nerves, endings of peripheral nerves, and the skin and mucous membranes. How is this infection transmitted? The mode of transmission of leprosy is not entirely known today. Scientists believe that it can be transmitted through respiratory droplets, direct contact, and it also can be transmitted by insects. Because Mycobacterium lepra grows slowly, incubation period of leprosy is very long. The average incubation period of leprosy is from 3 to 5 years. So in 3 to 5 years, a patient develops the first stage of leprosy. We call it indeterminate leprosy. When patients are in indeterminate leprosy phase, everything starts with a few innocent-looking lesions on the skin, the surface of the skin. They look like erythemas. They do not complain on the loss of nerve function, but actually if you get some tests and test those nerve endings, you'll see that there is a nerve damage already occurred. Then, if this stage of leprosy is not treated, it can develop into either tuberculoid leprosy or lepromatous leprosy. Let's talk about tuberculoid leprosy. It's called tuberculoid leprosy because the main symptom of this phase will be granulomas. And those granulomas look very similar to tuberculoid granulomas. Why immune system of that patient starts? And so patients develop granulomas on the surface of the skin because their immune system is irritated pretty badly by that time, and also they will actually complain that they lose ability to feel touch or temperature. So that means the nerve endings of those patients will be already damaged. So also patients might develop lepromatous leprosy, and this is the most severe stage of leprosy when patients become disfigured, blind, they start losing their fingers, toes. As I said, face will be disfigured, and actually, if you perform lepramin skin test on this phase of leprosy, this lepramin skin test will be negative. That means that immune system of those patients is completely destroyed. And by the way, lepramin skin test for tuberculoid leprosy is still positive. Treatment. Same as tuberculosis, leprosy requires long-term treatment. Usually takes a few months, and same as for tuberculosis, we're going to use combined or complex treatment when we use 2-3 antibacterial drugs at the same time. Why are we doing that? We're doing it to prevent development of resistance. And on this slide, you see a picture of a patient with leprosy, and patient probably in tuberculoid stage. And if you look closer at the arm, at this lesion on the arm, this is what we call actually granuloma.